Well, welcome, Mahinda, and we can hear you now.
Okay, great. Thank you, everybody. I'm going to talk about what I'm trying to do now with Physalis. It's a genus in which I have been interested for some time now, and I keep wondering, why are species close to the type locality easily recognized as that species? But once you start looking to other collections that go a little further away from the type collection, it's not so easy anymore. So what I'm trying to do is look with more detail into flower morphology, because as you all know, some species are very easily recognized for different reasons. For example, Philadelphica twists the anthers after the eyes. Solanaceus is the only one with ursulate corolla, and glutinosa has this very long tubular corolla. So those are pretty easy and straightforward and are easily recognized by everyone. But then you have what is more common in the genus, in which you have these rotate corollas that have spots that can be either solid blotch or composed from different spots, and color of the anther is also informative. But then you have this variation even in the same plant, which really puzzles me. For example, Physalis costomatal usually has large flowers with dark spots, and the spots are made out of different single units. But in the same plants, you will find corollas that don't have any spots. And then recently, with a recent trip to Baja California, I started looking into Physalis crassifolia, which I did not know from field collections. And there you have usually corollas with dark macula, and then I found some that are almost without a dark spot. They're mostly green. So all the the flowers I have seen so far all have a dense or not so dense tuft of hairs where the filaments get inserted. They have stomata in the ovary, which suggests they form nectar. Anthers mature at different days, and therefore they have different sizes. All the stigmas are wet and all have papilla, and the calyx grows in the fruit because it has a basal meristem in the flower, and therefore it keeps growing once the fruit matures. So this is some of the results I have been gathering with Ophelia and Pilar. They have this insertion of the stamens, and in the base of that, there's usually hairs. They can be very abundant and obscuring the insertion of the filament, or they can be almost lacking. This is one of the species that has very short hairs, but they are multicellular. Others are also very short. Then we have this stomata near the base of the ovary, which is present in all the species I have looked so far. I have only seen about 30 species so far, so I'm still missing a lot of taxa to look at. This is the stigma, and it varies from typically clavate, but some are more widely clavate than others without becoming capitate, but they are usually wet, and that's why you see all this trash going around the stigma. So these are the longitudinal sections of a whole flower with a larger area close to the ovary, which I think in some species might be a nectary, but which is almost lacking in others. For example, the one to the left has an enlarged area, which is lacking in the one to the right. The rest is pretty much the same, the petal insertion and the meristematic zone in the calyx. So which difference have I really found so far? Some ovaries stain very strong with neutral red, and therefore I am assuming they do have a well-developed nectary at the base. But others do have the stomata at the base of the ovary, but do not have the, they don't stain red with neutral red. So it's difficult to tell whether they are there or not. The corolla hairs at the insertion of the filaments can be, they vary. Some are branching, several indeed. Some are glandular, and some have mostly simple hairs, even when some branching hairs are also present. The stigma shape I have already mentioned. Some are very narrow, some are much wider. And well, I'm still working with that. I'm having problems with some species that are not so easy to cut. Some have a very large amount of crystals, and that makes it hard to cut. And what I'm working now with field work is gathering the genus north of the Tropic of Cancer in Mexico. The funding comes from CONABIO, and they're looking mostly for to form a seed bank that will be kept for their cultivation of the species. And the area is occupied by 11 states in the northern portion of Mexico. I'm looking into cultivated plants as well as native. And so far I have only been to Tamaulipas and Baja California Sur. And both states grow Physalis philadelphica. In Baja California only in a small area, but even when they did sow the seeds, they did not harvest because the price was not worth, and so they left the plants to rot. In Tamaulipas, on the other side, they have a very high technology cultivation. They had over 30 hectares of Physalis philadelphica, and they gather about 8 tons per hectare in this area. And what I find interesting is that areas that are not so well developed or don't have that much money to incorporate into the cultivation, what they do is they keep the plants without irrigation, even if it's the same species, Physalis philadelphica. They don't add water or fertilizers, and they keep the fruits small. And that's because there's another species, Physalis angulata, which is gathered from natural populations and is known locally as mil tomate. And people pay a higher price for this mil tomate. So what these persons are doing is trying to make the Physalis philadelphica look as if it was Physalis angulata, which is interesting because they don't even recognize that this is a different species. They just think it's a kind of different manipulation. So now that I have been trying different species, which I had never done before, I have been eating the fruits of the plants I'm gathering, and I was amazed to find that species that are not eaten in Mexico, Acerifolia, the one from the sandy beaches, Patulifolia, which you have in the United States also, and pubescence, which is frankly a widespread weed. They all have very nice tasting fruits. They are sweet, nice to eat. They don't leave any aftertaste in the mouth, but nobody eats them in Mexico. They don't even want to try. Once I said, oh, look, they taste nice, nobody wanted to try those. And well, some are very bad to eat. I can vouch that they are just horrible, like Crassifolia from Baja California and Nicandroides, which is also widely distributed. They taste terrible, and they leave an aftertaste in the mouth, which is not easy to get rid of. So that's all I have been doing now. If you have any questions, I think we have a lot of time. Thank you.
Thank you very much, Mahinda. Yes, if you have questions, you can turn on your microphone and just ask the questions directly. Or you can also use the chat and ask the questions through a chat. It's a very great presentation. I'm wondering also about I have one question about the variability in the leaf shape. Like you show that there is a lot of variability in the corollas, the color within the species, even in the same plant. And I think the same is happening with the leaf shape, that is very variable, very plastic within a species, and depending on the environment where it is found.
In size, they do vary a lot. In shape, I don't think they vary that much. And also with pubescence, you have these differences in which leaf pubescence can be very abundant and not so much in some others. But you can easily recognize some species by the leaves.
Okay. Well, thank you. Pilar, do you have a question? Hi, Pilar.
Hi. No, I just joined in this moment. I'm late. Sorry.
Don't worry, it's fine.
Hi, Mahinda. Great talk. It's Chelsea. So I was going to ask you, so in the beginning, you talked about like how the type specimens, or the individuals close to the type collection are very similar, but the further you get out, the more differences it has. I was wondering if you're looking at the breadth of the species, or are you kind of just going to the locality close to the type specimens to do your morphological work?
I didn't understand the question.
So in the beginning, you talked about you're going to the type collections, like it's really similar, close to where the types have been collected. Yes. I was wondering, because you are doing all this work with like the different trichome sizes and things like that, and like the stigma surface and things like that. Are you going back to like where the type collection was made, or are you kind of doing the breadth of the species?
No, I'm looking into a broader area of distribution. But
are you finding a lot of variation?
I don't have that many individuals yet. I'm trying to look at least at 3 different individuals, 3 different flowers from 3 different places, and I'm not at the point in which I can tell you there is this much variation. I'm still looking into it. For some species, I have only 1 collection so far. And so I cannot tell you yet how it's going to.
And ours.
Mahinda, it's Sandy here with Gloria and Franco in Córdoba. How are you? Very good to see you. Mahinda, I wondered about the nectaries, the absence of neutral red staining. So do you think the nectaries have been lost? Is there nectar in those flowers, or is it just, so what's happening there, do you think?
I think there is a very small amount of nectar in the flowers, mostly because I see bees in the field, which are gathering something, and then you look at the bees and it doesn't look like Solanaceous kind of pollen. So therefore I think they do visit the flowers looking for nectar. The thing is, I cannot find the placement of the nectaries, or they are just tiny because they don't stain with the neutral red, except for the very large flowers like glutinosa.
But maybe, so maybe they're offering some, because neutral, what is neutral red? I've forgotten what neutral red stains for.
Living cells.
Living cells.
Functioning cells.
So maybe, yeah, it would be interesting to kind of to grow them in the absence of pollinators and see if you get nectar at all in the base of those flowers. It's just really interesting that they would have that capacity but not develop, which is kind of, it's cool.
Well, I'm not sure which do and which don't, because if you look at the sections in the ovary, some ovaries have an enlarged area at the base, and that's very similar to what Nicotiana has, this very enlarged area close to the ovary. But so far I have only found that in maybe 6 species, but the rest do not have the enlarged area, but they do have the stomata.
Yeah, so I'm going on and on about this because I'm really interested, because solanums often have extra floral nectaries on the sepals and places like that, which are of course not the same as floral nectaries, but they are producing sugars. So maybe that's what's happening with the stomata on the ovary, and so it's not a nectary per se, like a sort of like a normal nectary, but it's something kind of quite different. That it's interesting because it could be an interesting synapomorphy or a loss, or, you know, as a morphological character. That's fascinating.
But then how do you recognize what they are producing? Do you make a chromatography, or?
Yeah, I think there's a paper that Greg Anderson wrote about extrafloral nectaries in solanum. It's a long time ago now, in the 1980s or maybe the early 1990s, and they stained for sugars, I think. I'm not sure. I'll try to find it, Mahinda, and I'll send it to you, okay?
Great.
But that'd be really interesting to look at.
Yes, because bees are looking for something, and I don't think it's pollen.
Franco, it's Franco now.
Well, I think maybe it's related to mating system. Probably the cultivated varieties have lost the capacity of producing nectar.
Maybe, but it's not just in the cultivated
selfers.
No, but it could be selfers. It could be selfers.
No, they are not. Philadelphica is a strictly open pollinated plant. If you don't have the bees, you don't get the fruits.
So there are no
strictly outgrowth.
Yeah. No, so it'd be interesting to see what the correlation is, because that way you could explore maybe what's happening. I'm not sure, but anyway, I think that's really interesting. Well, as a new, yeah.
Yeah, that's super interesting. And I also have a question, like, it's just something about the pollination. So it's more, these physalis are mostly visited by bees, or which other insects or visitors have you seen?
I have only seen the large European bee, the widespread European bee.
So mostly that. I think that Chelsea has been studying some pollination of some species like Physalis acucifolia. I don't know if she has something to add about visitors, or.
I would say it's true. I think Mahinda is right that they're looking for nectar, because what they'll do is they'll do that like little cute revolver pollination. So they'll like land on top of one of the anthers and put its proboscis up into the flower, and then it will just keep going around like kind of like a merry-go-round the flower looking for the nectar, and then it flies to a nectar. And I think that's pretty similar with native bees, but then you always get like your other things like your honeybees and your bumblebees and stuff like that. So yeah. But I do think it's like there's very little nectar that like I see even. So it's like, what are they going for? Because it just has to be like super concentrated or something for it to be a reward, because otherwise it's like, I don't know why they would be doing it. And also, I don't think it actually varies quite a bit from species to species, how much pollen is made and how easy the pollen just like is knocked off. And so some, it's just like totally like the bee will land on it and it be all covered with it. And then there's some like senescence is a little more like the pollen is like a little more into the anthers So I haven't watched too much of how that one's pollinated and how that might differ.
So syrphid flies never visit Physalis?
Flies? I have never seen a fly.
No.
And there was a suggestion by Darcy many years ago. He had a work on anthers, and he said that maybe bees were gathering crystals as part of the reward. And there are some species that have crystals in the anthers, but then some species do not. So that's not a constant character, at least in the species I have seen so far.
I'm not actually that familiar with crystals. What are crystals? Are they just like sugar compounds or?
No, crystals like calcium carbonate, or I think it's calcium carbonate.
Okay. It's calcium oxalate. It's calcium oxalate, and in Darcy's paper, what he said was that the calcium oxalate crystals aggregate around the dehiscence zone, and that's what makes the anthers dehisce. So all species should have at least some, because that's part of the mechanism of anther dehiscence.
Maybe they fall once the anther is open, because it's not universal that the crystals are present. At least in the ones I've seen so far.
So I don't know. It's interesting that you've never seen any native bees, Mahinda, because that's odd. Because I would have thought that there would be things like Melipona and have you caught them and sent them to the experts? Yeah, it's worth catching them. Because, yeah.
Hello. Can you hear me? Hello everyone, it's Andres here from Edinburgh. I have some reports from other bees in Physalis. I was recording some pollination syndromes for different groups in Solanaceae, and I remember some paper from Sullivan from 1984. He records some Alyptide bees, like some Bombus too, some Colletide too. And all these are native bees. Also some self-pollinated species like, let me check, Physalis angulata, Physalis heterophylla is also self-pollinated, Physalis pubescence. Yeah, it's not a lot of information, but it's some native bees recorded for Physalis.
Thank you, Andres. That's super interesting. Yes, yes, I think there's tons of work to do with pollination and visitors in Physalis, and also where they generally related. So that's super interesting. I don't know if you have any other questions. I know Isabella, Craig. Or Pilar, if you want to add something.
I have another question, not to get too stuck on these nectaries or anything, but also I feel like you talked about this, Mahinda, is that most of these flowers have these trichomes at the base of the filament, like the anthers within the flower. Do you think that's protecting the nectaries, or do you think
that was my first hypothesis, exactly, that they were always present because they were kind of getting the nectar in close to the base of the ovary. That was my hypothesis. But then, as you see in some of the micro photographs, those with the SEM, some hairs are so short and so close to the corolla that they are obviously not protecting anything. So maybe the long-haired stamens do have nectar and the short ones do not. I cannot say yet, but it's not, the hairs cannot protect, such short hairs cannot protect nectar, I would think.
I think it'd be fun to kind of talk about your second half of your project too, because we haven't talked too much about that, about cultivation of new species of Physalis. You kind of talk about that native people aren't super interested in eating new species. Why do you think that is? Do you think it's just a fear of something new, or?
I think so, yeah. The species that are usually consumed here are acid, which is the tomatillo and the mil tomate. They are acid and they are used as everything in Mexico with chile, so it's not sweet. And the ones I have been trying now from the north, those are sweet species, sweet fruits, and very similar to uchuba. I did like them a lot, and I tried to make the people that were working with me try them, and they just said, "Oh, no way, I'm not trying that." So not even the will to look into something different. I found that very interesting.
There's something similar. Oh, sorry.
Go ahead, Lucia.
Oh, something I was just commenting that something similar is happening in Argentina. Like we have the Physalis viscosa that is widespread in all the country, almost until the Patagonia. So, and the fruit is very tasty, sweet. It's a bit bitter, like the aftertaste, but it's very sweet. But no one is consuming. They're just cultivating Physalis peruviana mostly. And it will be interesting to see if they they start, I don't know, cultivating a bit the Physalis viscosa. It's a tiny plant anyway there.
Well, the spatulifolia is a very large plant, and the fruit is large, and it would be great for sandy, salty areas. So it would be a cultivation for very different areas than what is now under Physalis cultivation. But if the people are not trying to eat them, not willing to eat them, so it's not going to work.
Do you find that most of the orange ones are a little more sweet, and then the green ones is like the savory flavor or acid flavor?
So far, I have only tried 3, and they had yellow fruits except for pubescence, which has very nice colors because some turn purple and they taste nice.
Nice.
The other one that I tried, but it was in the US, it's Physalis fendleri. And the fruits are also orangey, very tasty, I remember. But most of them were infected by some insect. I can't remember the name. Probably there's someone here that can say something about the insects that. That's very variable between the species, right? Like there are species that are more, I know, like, yes, they're more with more insects sometimes within the fruits, and other that are more resistant, or
I don't know.
Hi, this is Joyce. We're working with Peruviana and Gracea and Prenosa, and we definitely see differences in insect predation on fruit and leaves between those. So we're starting to look at that a bit more. It's really interesting to see what's going on. So what was that? What was that, Ron Rocio, that you mentioned would seem to be really prone to insects?
Oh, Physalis fendleri is the one that was entirely infected. I remember that with Chelsea, we found one population that was a lot of individuals, but most of the fruits were very dry and all infected by an insect.
Okay. I'll have to get some of that. Okay.
Thanks. Okay, I don't know if there is any other question. No? Okay. If not, well, we can end this seminar. Thank you very much all for coming, and thank you, Mahinda, for the great talk and all the discussion. Next one, I think is not Chelsea. I don't remember. Chelsea, could you remind me who's it? Oh, okay. That's right.
We'll be talking about plant metabolic. Yeah, sorry. You can look at the website for more information about his talk. It's in 2 weeks. It'll be, I think, the second week of March. Sorry.
This is Craig. Can I just tease my talk briefly?
Yes, you can plug it.
Well, I think it'll complement this talk nicely. I'm a biochemist, so I'm interested in the chemical diversity within Physalis, but also all of Solanaceae. What I've found is sugars on the fruit surface in Philadelphia and potentially how they might get there. So I think it might connect really well with this talk.
Awesome. So we look forward to hearing from Craig in a little bit, in a couple weeks. Thanks again, Mahinda. It was really great to hear your talk.
Thanks for joining.
Okay, and thank you everyone. And remember that this talk will be available in the YouTube channel. You can watch it again and ask questions through this chat in any time you want. So thank you very much.
Great. Bye.